Hello, everyone, and thank you for joining us for this special discussion on neuroethics for novel neurotechnologies. And now I'd like to introduce Dr. Nir Lipsman and Dr. Judy Illes, who will introduce today's discussion and speakers. Dr. Nir Lipsman is a neurosurgeon and scientist at Sunnybrook Health Sciences Center and the University of Toronto. He is an assistant professor in the Department of Surgery at the University of Toronto and director of Sunnybrook's Harquail Center for Neuromodulation. Dr. Judy Illes is professor of neurology and Canada Research Chair in Neuroethics at University of British Columbia. She is director of Neuroethics Canada and faculty in the Center for Brain Health and the Vancouver Coastal Health Research Institute. Judy, Nir, the floor is yours.
Thank you very much, Jessica, and welcome everybody to this webinar. My name is Nir Lipsman, and I am in Toronto, and I will co-host this afternoon's session with Dr. Judy Illes, who is in Vancouver. As we begin, I would like to respectfully acknowledge that the land on which we gather, at least in Toronto, I know that we are gathering from everywhere, but in Toronto is in the traditional lands of the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples. We appreciate their land for this. We have an outstanding lineup of speakers this afternoon, including a very special plenary talk by Professor Helen Mayberg. This webinar has been very generously sponsored by the Focus Ultrasound Foundation, and we, along with the Pan-Canadian Neurotechnology Ethics Collaborative, thank you for joining us for the next 90 minutes. So I'll make some very brief introductory remarks before handing it over to Judy to introduce our speakers. Many of us who work and do research in the neurotechnology space, we recognize that our field is at an inflection point. So as our understanding of the brain continues to grow and evolve, so does our ability to intervene in it with rapid advances in imaging technology, both hardware and software. So we know that as brain diseases become more common and account for a much larger proportion of human morbidity and mortality, there is an urgent need to develop novel treatments and to better understand what drives these conditions. So in parallel, there is a growing need to evaluate critically how we investigate those treatments and to put the ethical questions in the appropriate broader context. So the field of neuroethics has grown from this and developed and rapidly expanded over the last 20 years and aims to address these questions really that exist at the interface between the brain and technology and to help prepare our field and ultimately our patients for these changes. So to lay the groundwork and to identify what some of the key questions are in the field and to try to anticipate where we're headed, we have formed the Pan-Canadian Neurotechnology Ethics Collaborative, which is a think tank of scholars spanning the neurosciences. And you'll hear from several members of PCNEC this afternoon. So Judy and I, along with Dr. McDonald, Pat McDonald in Vancouver, encourage everyone to join us on this journey as we chart new ground for this field. So on behalf of PCNEC and the FUS Foundation, we thank you for joining us. And I'll introduce Professor and Canada Research Chair in Neuroethics, Dr. Judy Illes, for some introductory comments.
Thank you so much, Dr. Lipsman. It's a great pleasure to be with all of you today. It's now just a few minutes after noon here in beautiful Vancouver, British Columbia. And I wish to acknowledge that the work that we do here and welcome you to is being conducted on the lands of the Musqueam, Squamish, and Tsleil-Waututh peoples, First Nations people. It's where Neuroethics Canada, our center at UBC, is located. And it's the location from which we have enormous privilege to conduct our work and share what we do with all of you around the world. I'm a neuroscientist by training, an imager, turned ethicist about 20 years ago. And all of that we are doing today in neuroethics is to proactively and pragmatically align solutions to thorny, challenging ethical problems with discoveries in neuroscience. And through this webinar today, we are pleased to bring you this content to our colleagues around the world to really consider neurotechnology and its implications for both people and society broadly defined, societies around the world. If you are inspired, if you have more questions, if you wish to contribute, please join us at PCNEC. It is a Canadian-led initiative through which we are embracing the international global community of doctors, ethicists, scholars, patients, policymakers, industry, and others. And we'll have a great deal of time for discussion after the presentations this afternoon to see what your thinking is, how PCNEC can help to serve our global community and really move the dial forward on functional neurosurgery and related issues around ethical and social and legal implications. And I too very much wish to thank the FUS Foundation for supporting us in this important endeavor. So let me introduce our three speakers now. They'll be speaking to you through a pre-recorded interview, a pre-recorded video, but they are here with us, of course, to join us in the Q&A that will take place a little later on in the webinar. So first is my very close colleague, Dr. Patrick McDonald, who's professor of neurosurgery at BC Children's Hospital and faculty in Neuroethics Canada. His research focuses on three principal areas: clinical outcomes and innovation in the treatment of pediatric syphilis, outcomes and novel therapeutics and imaging findings in concussion and mild traumatic brain injury, and bioethics and neuroethics. Neurosurgical implications that include conflicts of interest in neurosurgical research, ethical issues in fetal surgery for spina bifida, as well as pediatric epilepsy, as well as the role of ethics education in neurosurgery training. Dr. McDonald, thank you so much for being with us here today. Dr. Matthew Burke will be our second speaker and panelist. He's an associate scientist at Sunnybrook Health Sciences Center. Dr. Burke's research focuses on trying to understand and treat disorders at the interface between neurology and psychiatry. These include concussion and traumatic brain injury, functional neurological disorders, migraine, and other neuropsychiatric disorders. Dr. Burke, thank you as well. And our third pre-recorded video and panelist today is my dear friend and colleague in the neuroethics world and the health law world, Professor Jennifer Chandler. She's the Bertram Loeb Research Chair at the University of Ottawa. Professor Chandler's work focuses on the legal and ethical aspects of biomedical science and technology, with a focus on mental health law and policy, neuroethics, organ donation, and regenerative medicine. Thank you so much, Professor Chandler, for being with us today. And with those introductions in hand, I'd like our IT associates to queue up the videos, please.
Good afternoon, everyone. I'm Patrick McDonald. I'm a neurosurgeon at BC Children's Hospital and on faculty at Neuroethics Canada. I think it's quite fitting that we're beginning this series of small talks with the concept of vulnerable populations. I think the research and clinical care that we're talking about is almost entirely done on what we would consider to be vulnerable populations, almost by definition. And so I'm glad that I think it should underpin almost all of what we do at PCNEC. Vulnerability has been addressed either directly or obliquely in many of the codes of ethics we've relied on, certainly since the turn of the century. With that said, there isn't one definition of vulnerability that I think encompasses all that we need it to. And so I rely on a number of different definitions that serve a purpose for different aspects of vulnerability. The OED definition is the quality or state of being exposed to the possibility of being attacked or harmed, either physically or emotionally. Paul Ford at the Cleveland Clinic, in a paper he wrote specifically about vulnerability as it relates to neurosurgery, defined it as the circumstances wherein the established checks and balances of power and interest are no longer sufficient in promoting the just treatment of persons. And CMAJ, the Canadian Medical Association Journal, has actually made vulnerable populations one of their priorities, one of the things that they intend to focus on in their publications. And they define it as those vulnerable persons as those that experience adverse healthcare outcomes compared with the general population by virtue of both internal and external factors. Most of what we see written about in the ethics literature that speaks to vulnerable populations is in the research context. But if you look at when we encounter vulnerable populations, the vast majority of these encounters are in the context of day-to-day clinical care. So I think it's important to keep that in mind. The NIH has populations that they specifically consider to require additional protections, mainly being children, pregnant women, fetuses, and neonates, as well as prisoners. And although the CIHR or the Tri-Council Policy Statement doesn't define specific groups, perhaps other than First Nations, Inuit, and Métis, they define vulnerability as a diminished ability to fully safeguard one's own interests in the context of a specific research project. If you look at the populations that we encounter in the work we do at PCNEC, as I said, almost by definition, every patient we encounter is going to be vulnerable to some extent or another by virtue of having a serious neurologic illness. But I think we all know that those who have psychiatric illnesses are uniquely vulnerable. Children represent a uniquely vulnerable population. And I think in this context of what we do, disorders of consciousness and or capacity, whether that's a lack of capacity that's acquired or innate. One of the things that for me has really hit home, especially as it relates to the COVID pandemic, is the concept of what I call socioeconomic vulnerability. That's the vulnerability that comes about because of poverty, lack of education, geographic vulnerability, the vulnerability we see in racialized communities, as well as the LGBTQ two-spirit population, and I would add the transgender population to that also. For me, this has really hit quite close to home recently in Canada. Many of you may have heard of the disparities in vaccination rates in Toronto based on postal code. I grew up in the Jane and Finch area. I'd like to think that part of my ethical DNA is a result of growing up in that neighborhood and watching marginalized populations, whether it be because of poverty, race, lack of education, watching them really not being given the opportunity to take full advantage of the Canadian experience and what Canada has to offer. I saw this growing up, but it's really hit close to home with this significant difference in vaccination rates. Jane and Finch has one of the higher COVID rates and the lowest vaccination rate relative to some of the wealthier parts of Toronto. And we see this in access to some of the technologies that we study at PCNEC. We know that access to DBS in the African-American population is different than it is in the non-African-American population. Closer to home, Chris Honey has studied access to DBS in Canada and has found a significant geographic variability. The province you live in very much dictates how likely you are to get DBS. Interestingly, he didn't find a specific socioeconomic impairment to access, which I found a little bit surprising. And I suspect if we dove a little bit deeper into this, we would find that in Canada too. I think at PCNEC, there are, as I said, almost by definition, populations that we will encounter that are vulnerable. I would like some of the things we focus on to speak specifically to the socioeconomic vulnerability that we see in our country. I think it's important that in the research context, there's a fair distribution of risks and benefits of research, but perhaps even more importantly, once these technologies are widely available and accepted in the medical community, I think we need to play a role in ensuring that access does not depend on your educational status, how much you make, or where you live in the country. Much of what we will be studying and the patients that we treat suffer from treatment refractory illnesses almost by definition. And I think that creates a vulnerability and perhaps a greater likelihood to give into the therapeutic misconception. We know that in Canada, a vast country, that there are significant urban-rural divides in access to healthcare. And so I would like to see that as one of our emphasis. And again, perhaps unique to Canada, the role that First Nations, Inuit, and Métis persons play in our society, and the responsibility we all have towards reconciliation creates, I think, an imperative for us to ensure that they are part of this discussion. And I know Judy and Louise Harding are looking closely at that. One of the ways that we can do this is through vigorous patient or caregiver engagement process at the early stages of investigation, and perhaps even allowing patient and advocacy groups to participate in what we prioritize in importance in what we do. So that's just a whirlwind tour of vulnerability. Could have talked much longer on each of these subjects, and I just want to thank everyone for the opportunity to speak.
Thank you very much for inviting me to talk to you today about placebo effects in neurotechnologies. My name is Matthew Burke. I'm a cognitive neurologist based at the University of Toronto and the Sunnybrook Health Sciences Center. I'm also the placebo effects co-lead for the Pan-Canadian Neurotechnology Ethics Consortium. And so I'm very excited to talk to you about a topic I'm very passionate about. I think is very relevant to the development of many of the neurotechnologies that we're seeing today. So I have no disclosures. These are some of the funding sources for the research that I've done in this field. So I want to give a bit of context as to how I got interested in the field of placebo effects. I certainly did not go into neuroscience and neurology research with a plan to do placebo effects, and I did a cognitive neurology fellowship down in Boston at Harvard Medical School in Beth Israel with some great mentors here. And as part of that training, I focused on transcranial magnetic stimulation. And essentially, when I was reviewing different studies to put together this book chapter on clinical applications, one of the biggest things that I was, really struck by were that in many trials, not only was there, a large and notable active group response, but there'd be a very large placebo group response. So in this trial of treatment-resistant depression, you see this huge, whopping 37% remission rate of depression in the sham group alone. And there are certainly other TMS trials for depression where you see similar results. And despite these findings, a lot of the discussion would be about other points. I'm like, are we ignoring this huge, important topic here? We really need to, analyze and try and understand and interrogate this further. And the more I looked across different fields, the more kind of digging, the more interested I got, because in other fields like stroke and many others, you see similar responses sometimes where, again, here you see a whopping 2/3 response rate on their Fugl-Meyer score in this sham group. In this trial, the investigators couldn't explain this. They said, okay, there must be something active about our sham group, and actually ended up doing another trial with a different sham coil, but found similar results. So you can see how important this topic is when it comes to neurotechnologies, and that led me, really down the track of exploring this further. The thing that really bothered me the most, as I mentioned, is that there wasn't really too much conversation going on around placebo effects. And in general, in medicine, often the attitude is more of a negative rather than, okay, let's explore this further, let's understand it, and let's potentially leverage it. And so I joined the Harvard program in placebo studies, and Ted Kapczyk has been a great mentor to me in this field, and really have launched a line of research studying placebo effects. And placebo effects are a complex interaction between internal and external cues, and it surrounds the beneficial effects you're getting from the context surrounding a treatment rather than the treatment itself. So whether that be, caregiver interactions, care provider interactions, expectations, emotions, they all come together. And the keynote speaker today, Helen Mayberg, did some of the very important seminal work on understanding placebo effects in the brain, and showed that certain areas of the brain become activated or deactivated with placebo effects similar to antidepressant effect. And so this has been critical work, and many other fields, including Parkinson's disease and some other conditions, have been studying this, and it's kind of rapidly evolving. And more recent models show really network changes that happen in the brain with response to placebo effects. And when we try and think of what might be underlying some of those brain region and network changes, that's a whole topic on its own, whether it comes to positive expectation, reduction of anxiety, activation of reward centers. And I would guide the listeners to this great recent review article on this topic. Now, when it comes to clinical research, it's important to get a few terms out there. So when we have our classic clinical trial, part of the total response in the placebo group is placebo effects, but there's also other effects. And so you see that the placebo response, which is the overall response in the placebo group, is not just placebo effects, but there's other effects that go into it too. I won't go into details onto these, but it's important to delineate this, because the only way you could truly separate placebo effects from some of these other effects regarding, the effects of time and just being in a clinical trial and the observations, et cetera, would be having a no treatment control group. And unfortunately, these are rarely done in clinical studies. Now, when it comes to factors that affect the magnitude of placebo effect, there's a whole host involving the patient and the treatment. Given the scope of today's discussion, we're going to focus on the treatment, okay? And so this brings me back to one of the earliest clinical trials that was done on antihypertensives, where the 4-arm trial of antihypertensive pill versus antihypertensive IV therapy versus inert pill versus IV saline. So they have 2 placebo groups. And interestingly, unexpectedly, they saw that IV placebo was more effective than pill placebo at reducing hypertension. And so this was something that spurred a lot of discussion about placebo effects and intensiveness of treatment. It kind of makes intuitive sense that you might generate higher therapeutic expectations the more intensive or elaborate or innovative a treatment is. And so there's been a variety of research on this topic, but generally this is held. And so when we look at these brain stimulation technologies that are hugely elaborate, very exciting technologies, it's a huge issue that we need to be thinking about. And so there's many topics here that I want to just kind of give a teaser on. Obviously, given the time limits of this presentation, can't go into details, but just to highlight, how relevant it is to the topic of neurotechnologies and neuromodulation. So one of the critical components is you need to have appropriate and adequate blinding. And so the difficulty is you want to have a sham device that mimics the active device in all parameters, so the patients can't, know the difference between the two, but you want to avoid meaningful stimulation of the brain, and so that's not trivial. And for a variety of different technologies, they're working through this, but it's just critical to include a measure of blinding validity to see whether or not you are adequately blinding participants. There's issues of trial design here. Many in the pharmaceutical industry will know about the placebo run-in, where you're essentially trying to exclude high placebo responders. This is something that could be important in neuromodulation trials. It's a controversial topic. And again, further into trial design, outcome measures are critically important because they might be more or less amenable to be influenced by placebo effects. And our colleagues here have discussed important topics on this theme. And I'll just highlight one recent article that really emphasizes this. It was actually published a few years ago by Ted Kapchuk and some of the placebo researchers down in Boston, where they essentially showed when you had an objective measure in asthma, you see almost no placebo effects. This is a FEV1. Yet when you just switch the simple outcome measure to the subjective version, which is a subjective shortness of breath, you see maximal placebo effects, no different from the treatment. So critical to think about outcomes in fields where there might be high placebo effects. Then interpreting efficacy. So this was some work that we tackled on this topic, and I want to highlight one of the most important things that came out of that paper. When we look at clinical trials, a drug trial that maybe has a high specific effect and a modest placebo effect, if there's sufficient numbers and they're replicated, it might be significant, and regulatory approval might get, okay, green light. But if you have a device trial, which maybe has a larger placebo effect and more modest specific effect, in this type of trial, you wouldn't see a significant difference unless it was an extremely large trial, might not get regulatory approval and deemed, not effective. But when you look at the absolute improvement to the patient, which is what we should care about on some of these measures, you see that, it would be hard to argue that device B is at least, equivalent to drug A in terms of overall improvement. And, what happens when drug A is not available or drug A has lots of side effects? And it brings lots of interesting philosophical and ethical questions. Now, leveraging placebo effects is another topic. How do we learn from these large responses and inject them to improve the outcomes of our patients? This is a topic that I've been very interested in and talked, with the general media on this topic that I think has a lot more room to grow in medicine. How can we use neurotechnologies to better understand the neurobiology placebo effects themselves? These are, offer really interesting windows into brain physiology, and so we can use these technologies to better understand placebo effects. And finally, in this general field, we need to start thinking about the ethics. Can deception ever be justified? Because sometimes in placebo studies, you really, there might need to be some level of deception and informed consent. And, you'll see different tables like this going through different ethical principles. And it's important to think critically about research in this field and clinical work in this field. And so I think we're pretty much out of time. We've done some pilot work looking into this, these ethical questions. And in fact, in this small pilot trial, when we did a little bit of deception at informed consent, when we debriefed them and gave them the whole rationale about placebo effects while we're studying it, they were actually okay with it. So again, just early evolving work in this very complicated and interesting field. I'll leave it at that because I'm out of time, but I'll be excited to answer any questions in the panel session.
Good afternoon, everyone. I'm Jennifer Chandler. I'm a professor at the Faculty of Law at the University of Ottawa, and I am going to be speaking to you about selected regulatory issues arising related to neurotechnologies. Now, since I have 7 minutes, I'm going to be pretty speedy, covering over 4 main domains of potential issues. So we have the first area, which I've called device regulation, which is probably familiar to many of you, the various regulatory requirements, for example, by the US FDA for the licensing of medical devices would come up here. And I mention this here because as there's new strands of technological development, various regulatory bodies are asking themselves the questions about how should we go about regulating these particular devices. So just a couple of recent developments on the right-hand side of the slide, looking at how to regulate therapeutic use of extended reality, which might, for example, be integrated with neurofeedback, as well as more recently, a so-called leapfrog guidance for brain-computer interfaces. I would mention also that there are a range of emerging standards in development for brain-computer interfaces as well. So looking at the work from the OECD, the IEEE, the FDA, and European Commission all are discussing this right now. So as we start to see what comes out of that, that will become increasingly relevant, not just to product design standards, but will eventually probably inform regulation and also legal product liability questions. One other issue that comes up here with regulation has been this question of post-trial responsibilities to participants in invasive trials of invasive devices. And the issues that are coming up there are whether there's an obligation to cover costs of explantation of the device, or conversely, to facilitate continued access to supply replacement batteries. And this raises questions about who's going to pick up the ongoing costs: the researchers, the device manufacturers who sponsor the trials, or publicly funded healthcare systems or privately funded insurance schemes. So I won't say more there, but just mention that there's this whole zone of device regulatory questions related to neurotechnological developments. The second topic I wanted to mention is that of clinical regulation. And here we can distinguish a couple of domains of clinical regulation. And one is specific regulation of certain neurotechnological interventions. So NPD here stands for neurosurgery for psychiatric devices, formerly called psychosurgery. And along the right-hand side, you'll see a recent consensus guideline coming from the clinical field, a response by a functional neurosurgeon saying, hey, we actually need laws, not just guidelines. And at the bottom, a recently published January 2021 paper that I wrote along with 20 international co-authors functional neurosurgeons, ethicists, lawyers, looking at international laws or laws from a range of jurisdictions around the world governing neurosurgery for psychiatric disorders. And this is interesting because the definition of NPD in these laws varies quite a bit and actually can be quite a bit broader than you might think. For example, in Queensland, there's specific rules relating to non-ablative neurosurgical procedures, which are defined as procedures on the brain that don't involve deliberate damage to or removal of brain tissue for the treatment of mental illness. And this could conceivably include quite a wide range of non-invasive forms of intervention. DBS, deep brain stimulation, is often captured by these laws, and Scotland specifically regulates transcranial magnetic stimulation. So I offer this by way of an example of the existing specific, technology-specific and indication-specific laws that exist around the world. Also, of course, there's general laws of general application that apply to all clinical interventions and have to do with the standard of care applicable to new and emerging treatments. So there's a whole range of issues of medical law to consider there, as well as research regulation. The third topic, which I've identified for this selective overview, is this question of access issues. And so here we're looking at questions of the cost, funding models, and the accessibility of the technology to patients. And this interacts with regulatory questions. So there's a little example of a paper from one of my colleagues in PCNEC who wrote about the regulatory approval of a new device which had a shortened battery life without evidence of therapeutic improvement, leading physicians to switch to the use of rechargeable devices so as not to expose their patients to repeated surgeries for battery replacement. But of course, the rechargeable device was more expensive. So this regulatory approval of the new device led to the use of a more expensive device, which has knock-on effects for the availability of the device in general to patients. If people are switched to a more expensive one within an environment of constrained resources, fewer people will have access. There is also questions around exclusion of patients with cognitive impairments from certain forms of long-term DBS therapy or replacement of batteries, which raises questions of human rights law and the kinds of arguments that might be made around access to treatments. Medical tourism is another issue that's arising with respect to brain interventions, and this is a reflection of whether technologies are available, but also of their cost in a given jurisdiction. So one can see online that DBS is widely advertised to medical tourists in high-cost jurisdictions. And this raises regulatory issues as well about what to do with people who are exposed to unsafe treatments or who come home with difficulties, the cost of which then has to be assumed by the domestic system, but also raises issues about displacement of care in the destination jurisdictions where people go to seek these treatments. And the last issue I wanted to raise in my short overview here is this question of intellectual property. And so this is a domain of law dealing with rights to intellectual or creative products. And we can look at issues in both patents and also copyright being relevant here to neurotechnologies and developing neurotechnologies. So along the right-hand side are a couple of references to articles, including by colleagues at PCNEC, Zelma Kiss and Judy Illes and colleagues, looking at the increase in the number of method patents being filed for stimulation or neuromodulation, I should say, of specific brain regions for certain conditions. And the concern here is with the accumulation of overly broad patents that might impede further development or research in the area or development of other products. And this kind of concern about overly broad patenting is reflected in the whole saga of myriad genetics and the BRCA gene patents. And questions are raised about the breadth of the patents that are being offered, as well as whether specific brain zones should be eligible for method patents. Another issue which I see emerging, but it's rather hard to document, is the impact of copyrights which are the kinds of protections offered to software arising in relation to neurotechnologies. So if we can get our hands on, which occasionally we can, find copies of the end user license agreements or the software license agreements under copyright law that that govern the use of software that might be integrated within a closed loop implantable medical device or within programming devices for those implanted medical devices. And these copyright end user license agreements, not usually available to the patient because they tend to be that information tends to go to the hospital or the clinician doing the implant. But they typically include limitations on reverse engineering, decompilation and disassembly, disclaimers of warranty, waivers of liability, and sometimes include a consent to the collection, use, and use of data. And I think as we're moving to more closed loop forms of implanted medical devices, the ability of a patient to have access to an algorithm, especially if it's an adaptive kind of closed loop device, becomes more important. And especially if we look at closed loop devices that modulate important cognitive functions. So I think this is an issue going forward to have a look at what forms of restrictions are appropriate to recognize and permit within copyright license agreements that ride along with these medical devices. So I'll stop there and I look forward to the rest of the discussion. Thank you so much.
Thank you so much, Patrick, Matt, and Jennifer, for those outstanding introductions. So those represent three themes amongst several others that begin to describe some of the challenges facing the interface of technology and neuroscience. Just as a reminder, we are going to have a dedicated period of Q&A following Dr. Mayberg's talk, so there will be a chance to ask any of the speakers any of the questions that you might have. So at this time, I'd like to introduce Helen Mayberg, who is one of the true pioneers of neuromodulation in psychiatry. Dr. Mayberg attended medical school at the University of Southern California and then completed her residency at the Neurological Institute of New York at Columbia University. She has made seminal contributions to her understanding of depression, including a critical pivot toward circuit-based understanding of the disease. So following positions at the University of Toronto, where she led the team performing the world's first trials of DBS and depression, and at Emory, she is now the director of the Center for Advanced Circuit Therapeutics at Mount Sinai in New York City. So her achievements are numerous. She is a member of the National Academy of Medicine, the American Academy of Art and Science, and the National Academy of Inventors. And we are just delighted and privileged to welcome her as today's plenary speaker. Helen, over to you.
Thank you. What I want to do is take the opportunity to kind of frame a neuroethical issue, but really not as a neuroethicist, but what I think 20 years now almost of thinking about this problem of DBS for depression has led me to focus on when I think about expectation. And at the end of the day, if we don't manage expectation, our expectation as scientists, the expectations of our patients, our research subjects, we will continue to have problems as we advance with more novel and unknown neurotechnologies. So I see this as an optimistic talk, but we'll try to share some of the things that perhaps we've learned the hard way briefly. And then we hopefully can discuss many of the things that have come up in the fantastic introductory talks. I have some disclosures. All of this is in some ways unfortunate, the ongoing off-label use of some commercially available devices, but now really prototype devices. They're all registered for study now at the FDA. The funding is mostly now from the NIH, and referable to the last comment about intellectual property, this initial work from Toronto did lead to a use patent that I'm happy to discuss. Is that good or bad in terms of thinking about innovation, access, and competition? I think the biggest construct, and I want to put a little neuroscience back into this is not just for depression. Depression is a use case, but neuromodulation generally has a construct that we are treating circuit disorders, which implies that what we are doing has a fundamental biology. And if we just work hard enough, understand the details, utilize all of the scientific advances, we can reduce these very complex neuropsychiatric disorders down to some fundamental principles and targets where we can poke it, stimulate it, block it, enhance it, and do good things. And I think that is an organizing principle, and we can discuss whether or not that is an oversimplification. The other thing that I think is important is that regardless of whether or not you have a novel, non-invasive device, a intermediate kind of device like focus ultrasound, really appreciating that this is sponsored by the Focus Ultrasound Foundation, where one can use their technology for both ablative purposes, but hopefully in the future, some very inventive neuromodulatory approaches. And then, kind of the workhorse, deep brain stimulation. Regardless of how we do this, we have to know why we need it. Whether even a non-invasive technique requires going to a doctor, having a device, having costs. So why do we need it? What are we trying to do? The second is, well, where are we going to stimulate? And that obviously isn't so much an ethical issue, but is a very pragmatic issue that has to be addressed with every new technology and every new indication. We ought to have some idea of what should happen. And I think fundamentally that is an ethical consideration, because that's where the mismatch of expectation and reality really meets. And along the way, vulnerable populations, of which many of these treatment-resistant patients are, is who is eligible. And as you have devices that are expensive, high maintenance, need to be followed along with access to scientists, engineers, clinicians, who is eligible and how do we ensure parity and access for all those in need? And I think that to those four main pillars, we need to determine if circuits can be safely and effectively modulated. And that really is fundamentally my job. We need to figure out if we can match a target in the brain to a particular disease, a syndrome, a symptom, and then a patient. And that really is fundamentally what we think about all the time. Are we tuning illness circuits? Is it that we're only going at particular symptoms and we may do a great job of changing a particular behavior and actually do nothing to a patient's well-being or even to the syndrome for which the symptom lives? And if all things actually come to bear, that we can devise personalized approaches that optimize response and get the best option for a given patient. And this is really a mantra, whether you're trying to decide between drug and therapy, or you're trying to decide between different targets with DBS, or you're trying to distinguish between different kinds of invasive or non-invasive procedures. So if we actually get to the use case, I think it's really fundamental as we get into different kinds of technologies, different kind of biological processes, that we get back and never forget the patient that we're dealing with. And I find that over the years, actually listening and having patients describe what their experience is and to have them understand what they want to change is really a centering principle to do this work every day. And as you can read from one of our relatively recent cases from Emory, that depression, whether it's treatment resistant, which only adds insult to injury, is the fact that depression is not an episodic problem, but actually is an all-enveloping state. And the idea that your brain can hijack every aspect of your being, whether it's moving or thinking. The idea that patients describe feeling paralyzed, which obviously they're not, and that the process of moving to get something done in a day actually takes aim at their identity, is a combination of behavioral elements that in many ways defies simple brain organization. So we have to ask ourselves as we enter into treating people with these kinds of conditions is, what might recovery look like? And what is it that recovery should look like? Is it to relieve pain? Is it to make it so people feel they can move? Is a return of agency really the primary mission? And perhaps one would be hard pressed to say you could only choose one, but I would like to offer the idea that the order of what changes may be fundamentally important. So I think the other thing to really keep in mind as all of these technologies are entertained is that in depression, when one thinks about access, when and who, by the time you've failed, or by the time you've had 5 episodes, even if you've recovered previously, the likelihood by the time you get past 5 episodes that you will stay well outside of a year drops down to almost, immeasurable levels. And so our bar, whether it was what we were thinking, Sid Kennedy, Andres Lozano, and I so many years ago, was that who do you offer an unproven, invasive brain surgery? You have to consider it's in people without any options. But by definition, the more options that you have failed, the likelihood that some new one that you design will be effective is really a pretty bold assumption based on the evidence. But that in the absence of anything else, then the question is what can be done safely and with a justifiable rationale. And I think that when you have a disorder where even since the initial studies of DBS for depression, there have been 3 approved treatments, but none of them actually are reliable or tested for patients who have failed ECTs. So there is a niche. Thankfully, is not a majority of patients with depression, but this is the group in which we have focused our attention. So as people know and can read, and it's been around a long time, this was an approach to basically target an area in the brain that seemed to be linked to successful antidepressant treatment, as well as at the core of the experience of extreme negative emotion and subsequently pain, but that over time, targeting this area with high frequency stimulation in the subcallosal cingulate area 25 led to actually an unexpected, and I will say, and you can take it or not, a really remarkable sustained effect in many patients who have actually been implanted. The experience at Emory, which now exceeds, this is an old slide, probably more than 200 patient years. And even though it's a small cohort of people who've been continually studied over this period of time by the research team, that actually if you get better, you stay better. And this had been the experience in Toronto of the original cohort, and even in failed clinical trials like Broaden, those patients that did well with long-term follow-up exceeding 5 years can continue to achieve and maintain benefit. I think the idea that in an illness that is notorious for if you can get better, you relapse, the latest treatment, ketamine, needs to be repeated and people will relapse, is the idea that this does behave like a pacemaker, that the brain is altered, it clearly is not recovered, but actually you can maintain an effect where previously an effect could not be achieved. Now, the technology now has advanced. Area 25 may have been first and does have the largest number of published studies by various groups, but other targets are certainly yielding very interesting results. But whether it's unilateral, bilateral stim, people are entertaining multiple targets, whether or not it's open loop with high frequency stimulation, whether it's using local field potentials to define parameters, whether or not it's trying to move to closed loop, all of that is underway to kind of make what seems like it is working, but isn't working well enough to be scaled up. There are lots of options and they are more and more evidence-based. There's even new work now as typified by the group at UCSF where you treat a depressed patient like an epilepsy patient. You put in multiple stereo EEG leads invasively in the brain. You monitor for a number of weeks in the hospital. You look for a personalized mood readout, and then patients come back for another surgery after the data has been analyzed and have a patient-specific implant that actually reads their individual mood and actually interrupts it as it comes online based on the brain signal. Now, the times are incredibly exciting, but the bottom line is none of these approaches, including the ones that we've worked on for so many years, have reached a commercial threshold. People are now, debating the best design. Should people have access based on the evidence to date? But I think beyond that, for our discussion today is, with all of these trials available and people, well, before COVID, traveling to be in trials, or even teams offering multiple trials because you had scientific expertise and access to different technologies. How do patients choose a trial? More importantly, how do doctors prioritize their science, and how do they stratify patients among these options if they have more than one? And I think that would be fun to discuss later. I think the other thing that's important to the idea of my theme on changing expectation is that we had always assumed that treatment would develop over time for depression. We didn't yet appreciate ketamine, that actually treatments could give very, very rapid and almost immediate effects, but we actually saw this in real time in awake patients in the operating room, where they came to the operation with any one of a number of really overwhelming metaphors to describe the state they're in. I've just kind of put a few that people have described over time. And when you actually are in the right place and apply generally a threshold of current at high frequency, patients have an absolute immediate shift. And this shift is now in our hands, reproducible, predictable, based on tractography. And we can know in advance whether it's blinded to the person who's watching the patient and to the patient, but that we can know in millimeter precision which contact is going to basically reset or turn off the high negative affect. What we never really thought when we saw that was an antidepressant effect, but it is quite a stunning experience for patients, which does set a stage for them for what their recovery will look like. And the reality, which should be no surprise for any clinician, is that you don't get depressed like you have a stroke where it comes on suddenly. You equally don't recover from depression as though you just turn off negative pain in your habits, your biases, all the behaviors that have been part of your depressive illness don't magically disappear either. And then what the patients come to realize is that there's a lot of work involved in the recovery process. So from a tech point of view, we need differential metrics to actually understand
this time course, which is extremely interesting. From an ethical point of view, patients need to suddenly be reframed. What their experience is, what their expectations would be, it's almost as though they are behind a curtain. They cannot see what is possible until they get out from behind that. And there's no way you can explain what's going to happen to someone prior to surgery. So things change once those initial symptoms abate, and a patient, you cannot frame it for them in advance, and they can't know it until it happens. So that does lead to looking at what happens over time, and it is not the same in individuals. That initial effect may be predictable, and the experience is similar but not identical for different patients. But the timeline over the months of whatever a trial is, but over the course of the first year is really variable. And so what we need now is we use the same settings for all phases, but there is this variable response rate, which is the focus of a lot of scientific study. We need biomarkers that can track. We need readouts of the brain and behavior to know when to adjust. We need to, importantly, we've learned, discriminate habituation, needing to turn the stimulator up, from relapse, of which one needs to jump on that, versus moment-to-moment fluctuations and life stress, which actually may not need a device adjustment at all. And I think importantly, and more existentially to think about for the patient, patients are having a change in their perceived experience as their brain is changing. So as we're trying to figure out how the brain is changing, we're watching a patient's predictions about their behavior change. And that's actually a very, very interesting computational psychiatry question, but it actually wreaks havoc on the course of a trial. And it actually makes it difficult for a patient to actually understand what's going on. And that is a work in progress. I think we need readouts that are also so that we can mitigate risk, that we need sustainability. We heard about, how do patients relate to batteries? What if you can't get a battery replaced? Having some knowledge about what happens if your device breaks or your battery isn't replaced in a timely manner. Are you stimulated as in Parkinson's? You turn that device off and in 2 minutes, your symptoms are back. If you are stimulating the anterior limb of the internal capsule, like is done for OCD or in some depressed patients in the monoaminergic pathway, if you turn the device off, patients go into a profound change in negative mood within days. In our target, it looks like it's a slow slide, that you actually have a window that we've had a lot of experience with batteries wearing out and needing to be replaced with controlled blinded discontinuation. You don't see symptoms generally over the course of a week. And even with battery failures, we've learned that you can recapture the effects. So it's important to know, are you doing something that has a real safety window, and that can be studied and has to be considered. And that becomes very important for patient empowerment and agency over having a device, knowing where they can get into trouble and when their own safety window is. We are moving toward how can we develop these technologies so the doctor doesn't have to be in the loop, and that the patient has more control and there's more reliability. And that's, in the short term, tremendously labor intensive, but hopefully in the long term will lead to better and more scalable and accessible treatments. And our own work is really trying to say, can we get beyond self-report? Can we have readouts that are reliable surrogates of how the brain is recovering that we can use? And this can get incredibly elaborate. There's now a bragging right about how complicated your experiments are. We now have, with our NIH study, access to the Medtronic platform that allows you allows the patient to actually turn on the device at home, collect the information. We're doing video, actigraphy, multiple recordings a day, logging on, having patients do depression ratings. We're trying to be rigorous. We're trying to take advantage of these next gen devices for their novelty and their opportunity. But we're talking now about much more limited access and tremendous burden, and we have to balance all of these things. I want to kind of just end with these last two slides that really hopefully will frame our discussion, is that, look, the brain is not a bone any more than, the heart is the brain. The brain is, so complicated and is always changing. But I think it's useful to consider some simplicity that when an organ is broken, when a bone is broken, the first thing is you have to reset it. And once it's reset, doesn't mean it's ready to run the marathon, that there is a period of remodeling that actually may seem as though there's more constraints than there were before it was broken, but that even when the brain is, or the bone has reached a stage that it's ready to retrain, the rehabilitation process goes in steps. And that is generally not done by just going out and doing what you've done before. Your bone and your body is never the same after a break. Your brain is not the same with exposure to an illness, whether it's depression or another. It's been remodeled, and that as you have a device that is helping it to repair, that you need to learn what your new brain does. And that's a process. So what do we want? We want patients to be better. We want them to be better. And importantly, with these invasive devices, if they don't stay better, we're doing something wrong and we need to reconsider. But I think now, not only do we need to understand the devices and to use the ones that are best, but we need rehabilitation strategies that maximize recovery and resilience. And that may be different for different devices and different strategies. But the patients can't be passive partners. And that's part of this too, that patients need to learn the difference between being distressed and being depressed. And maybe we can help them by thinking about readouts that can tell the difference. And I think as I show, my team members here, I don't have a picture of Peter Jacoby, who worked with Sid Kennedy on our cohort in Toronto and still continues to see some of those early cases, is that when you do investigational studies, it requires ongoing, long-term, and what I mean by long-term is indefinite commitment and a relationship with the patient, with the team. And that is not something scientifically we think about. And I think everyone needs to remember that as we want access and we want to make these things accessible, that we all have a responsibility to the patients and to the science as you take this kind of work on. So I think for thinking about, the next new thing, and I think this is what we're here to discuss, for patients, look, recovery is more than a stimulator. We have to manage over time therapeutic misconception, and it just keeps changing. And it's not just for the patient, it's for the family, the referring physicians, and actually the community. We need, biologically based reframing of expectations, because expectations is a fancy way of saying, brain predictions. And there's a biology of brain predictions, and we are right in the system of the brain that actually does that computation. So we need to figure out how we retrain people while their brain is being retrained. I think we have to anticipate and manage future unknowns. And I know that sounds like an oxymoron, but things will happen that we don't know what they are till they happen. So we've got to be prepared. And I think we do that by treating patients as collaborators. So I think considerations for discussion for all novel interventions, you actually have to plan for both good and bad outcomes. I can say very honestly that when we did those first patients in Toronto, I actually designed the experiment. I wouldn't have done it if I didn't think it wasn't logical and the science didn't point us to our target, but I actually designed the experiment to know that when it didn't work, that I'd know why. I actually never really did the mental computation of what I would do if it did work and how we would think about taking care of these patients long-term. And that has been the sobering reality that I warn everybody in this field to remember. I think that all of this sets the stage that we need creative and unique trial design for devices, but that can be debated. And I think importantly, we have to have long-term stewardship, particularly when we use prototype devices that may not have commercial backing. And we need to think very seriously about what you will do if you have a prototype that works, how you will maintain it for patients if they do well. And I think that does bring up the ideas, when does an experiment become a treatment? So I'll just leave you back with something from one of my patients, because patients get it, and you just actually have to ask them. And that's what we always have to do as physicians. I think we lose sight of it in blinded trials where nobody talks because you're so afraid that you can influence patients. You can't influence them long term. But as this patient said, I have a lot of learning to do. I sometimes feel quite lost, and none of this is anything like I went through all those years. I'm just trying to figure out who I am and where I'm headed. I'm happy, and I'm definitely overwhelmed, but I'm not sick. And I think trust patients to actually help you to know what is the thing you're treating. And it's for us to help the patients to understand those things that we're not treating. And I'll stop there.
Hi, everybody. And I want to thank you all for those very informative and thought-provoking lectures. They were really outstanding. I clearly see how it takes a village of ethicists and legal experts and scientists and clinicians to all work together in this emerging field. Dr. Mayberg, your presentation was outstanding and identified so many exciting issues and also so many, for me, fearful issues of how we can go about doing this carefully while maintaining and ensuring the safe and efficacious delivery of care to all of our patients. So thank you all. Thank you. I think has all the panelists turned their cameras on, as well as some new people that may be able to turn their cameras on. Let's see. Oh, okay, let's see if from some other people. And for the audience, please submit your questions via the Q&A function, and we'll try to get most of them answered. Okay, so let's perhaps start with a question for Dr. McDonald, and all the panelists, please feel free to address them. I'm so interested in hearing your back and forth discussions. It seems as though there's heightened desperation for access to new therapies in some vulnerable populations. How is the risk of like an optimism bias in participants managed in the case of clinical trials for new therapeutic devices? How do you manage that?
That's a great question. And I think Dr. Mayberg's ending about the importance of the therapeutic misconception is critical to managing those expectations. Expectation management, especially in the early development of any treatment, whether it's a device or a new pharmaceutical, is critical. Even when you do that, it's hard, especially for a patient who is kind of, or a family that's at the end of their rope and has no options. It's very difficult to manage that. And I think you have to continually reevaluate the patient, continually reinform them that we don't know if this is going to work. I think several of the speakers have talked about the importance of involving patient populations in the development of trial design and things like this. And I think that might be one of the ways that we might get a little better at expectation management is we focus on patient involvement and patient engagement in the earliest of stages of developing these new treatments. Someone who has no hope will look for hope anywhere, and we don't. It's a tension between taking away that hope and managing expectations. I don't have an answer that would take care of all of it.
I might kind of add that what's interesting, when, with the long history of this kind of not yet approved depression treatment with DBS is that, look, patients read. They can read, long-term follow-up studies. And, trying to come up with a ethical equipoise personally of when people are screened to actually say, we think we know based on experience who's an appropriate patient for this. You meet that criteria as we've defined it. We know how to implant, surgeons experienced. We know how to make adjustments. What we do not know is who we shouldn't operate on, even having met all the criteria. So one way to not, say something that just doesn't pass the smell test of, well, we really have no idea if this works or not because it hasn't been commercialized. So, I just kind of throw that out as what I found, that if patients know that you're committed to doing something, even if it may not work for them, then that's one framing that can be helpful. But I think when we get into these brand new treatments, what is it? I find that, we get a lot of calls from patients that are shopping DBS trials. There are very few of them, but they're around. And if people have means to travel, they're trying to figure out, and it's like you're being interviewed. And, again, it's people, you have criteria, and people need to qualify, and they need to make decisions if they're appropriate. But it's not a negotiation, it's a study.
Yeah, and I would just highlight, for the question regarding optimism and expectation, these are core pillars when it comes to placebo effects and placebo responses, and it really depends on the context too. In a clinical trial, it's obviously important to maybe subdue that a bit, or else you're shooting yourself in the foot in terms of the optimism and expectation. Obviously, in clinical practice, it's much more challenging, because if you were to do that, which might make things, more realistic, but then you might lose out on some of the placebo effects, which we're, learning more and more does biologically change the brain. So if you can get an additional effect in there, well, that's important too. So it's really controversial.
Suzanne, I'm wondering if Dr. Kish in Calgary would like to say a few words with respect to optimism bias and some of the discussion we're having. And then I'd like to jump in with a question for you when there's an opportunity.
Sure. Dr. Kish.
I thought there was too many people on the panel. That's why I was kind of hiding. I'm not quite sure what more to add to what Helen just said. I think that, I agree completely. We've got lots of people out there who are ill, who are looking for therapy, who aren't getting the therapies the way they want them. And so they do tend to read too much. And maybe we are doing a lot more, it's the availability. It's the availability of the information to the general public. And so you certainly can get a self-selected group that might not be your ideal candidates for the studies.
In the clinical ethics world, we saw a move, like a shift of the pendulum from left to right, and now a little bit more to the center from, full-on paternalism, many decades ago or up 2 decades ago, and a swing all the way over to what I would call consumer-directed care. Patients actually demanding therapies, drugs, interventions, all kinds of things based on their, new access to the internet and the push to have them, assert their autonomy, and as people with certain kinds of expertise in the disease areas that they have. And I think that, the ethics literature anyway suggested, and the data showed that sort of polarization of consumer directed intervention was really not beneficial, maximally beneficial to patients, and a little bit of swing back to the more centered, a bit more shared decision making was at the end of the day had better outcomes for patients than these sort of polar paternalism or public directed or patient directed care.
Yeah, I find that interesting because with advertisements on TV and patients learning about studies or drugs from the television, sometimes the physicians aren't aware of all of these new drugs that have been approved or new technologies that are available. So I think the patients can bring it back to the physician for discussion, and as you said, have it be an intelligent conversation between both of them. But I never cease to learn from my patients and vice versa. So I welcome the knowledge, but I put it in perspective. What about you, Dr. Lipsman?
Yeah, no, I agree with that. I also had a question for Helen and the other panelists about sustainability, and the idea of now with depression patients in some places being implanted with SEEGs and treating them with the same way we treat epilepsy patients maybe, and how do we balance sort of the costs of neuromodulation with the data obtained and with trying to find data that is sufficiently impactful to make a difference on people's day-to-day life. I mean, I think we have a very strong interest in Toronto on outcomes and on redefining how we evaluate outcomes after neuromodulation. I mean, the last slide that you showed specifically with the patient's quote, I mean, in some trials, that patient would be maybe a non-responder, but clearly they recognize inherently that they're no longer sick. And are we capturing that properly? So I guess it's a two-part question. One relates to sustainability. The other is, are we, and I know you've spoken about this in the past, but what risks are there if we don't capture outcomes accurately?
Well, I think, in order to mitigate, I'll say the last part first. So first of all, that patient actually was in remission. So talk about personal insight, and that she can really discriminate, being what is moment to moment chaos in her life versus this is not being ill. But that is a woman that actually was able to do that. And you speak exactly to the fact that some patients are doing things, look better, are totally transformed from who they are, and will insist that they are no different. It's almost like anosognotic for their recovery. It's I don't know what that is. It needs philosopher, and it needs behavioral neuroscience in a way that I'm certainly not equipped. But it is a phenomena. I think we equally have to be careful not to chase after this holy grail of what other metric we should use. But I will make the analogy to in Parkinson's. They certainly don't use the entire UPDRS, all the scales, to decide if dorsolateral STN is effective for motor symptoms. They cherry pick what they do from their own scales because they actually have the neuroscience backing to say, you're in the motor system. It certainly, I would hate to see in Parkinson's if you were judged by whether or not you treated Parkinsonian apathy or depression or impulsivity, and your failure to do that meant you weren't entitled to actually not give DBS to alleviate tremor and rigidity and bradykinesia. So I think that psychiatry indications suffer from we don't even buy our own measures and we don't have a readout. So I think that, to the sustainability and looking at the SEEG approach, on one hand, it seems really appropriate because, hey, it can be a biologically based biomarker. You can argue that's kind of overkill maybe, and maybe you didn't need to do SEEG. Time will tell. We'll wait to see the publications on that data with more than a couple of case reports. But it does beg the question of, if you develop a biomarker to follow, it may not be the right one. And I certainly am of the opinion watching patients now that you can have depression as a binary and actually be fundamentally better and have overriding on that the noise of everyday life. And some people's everyday life is much more reactive than other people. And I would defer, as a non-psychiatrist, to Peter Jacoby, who's on, to comment on that. And so there's multiple rhythms. There may be a depression rhythm that we can get a readout on, and we know that you take that part of the syndrome away and you're done. And then, but you got to know what you are treating as well as what you aren't treating. And I think that's part of this expectation. I think we're kind of still balancing out which one we pay closest attention to.
Thank you for those comments. And I think Dr. Jacoby is on and can address some of that. Hi, Peter.
There. Sure, I'd be happy to share some thoughts on that. I think what we often tell patients is that the goal of treatment is to make you antidepressed and may not necessarily make you happy. I think that's a different sort of outcome that comes from life events and connections that you have. So, as a way of reestablishing homeostasis, we can treat those core symptoms and turn off that signal that we're talking about. Does that ultimately lead to other outcomes that are more based on happiness? I think that's where the rehabilitation comes in, that Helen mentioned, and to get people to those different outcomes requires a different set of interventions.
Yeah. I think we also have Dr. Fasano on, who maybe can relate his experiences with the Parkinson's comments. Dr. Fasano on? No? No? Okay.
Suzanne, can I jump in with a question for the group? So I'm really interested, I want to take a shift to looking at the future of the Pan-Canadian Neurotechnology Ethics Consortium and what we need to be thinking about in the future in leading the way, both in terms of neuromodulatory technology and ethics. And I'm wondering what all of you think about the future of open science and its intersection with neuromodulatory techniques. So we're seeing a movement towards data sharing. So, outside of the realm of open access and so forth, but really access to patient data. In Canada, there's a big push now to, led by the people at Guy Rouleau and others, the Montreal Neurological Institute, to move away from patents. Jen Chandler talked a little bit about patents in her presentation. What do we need to be thinking about in terms of open science? I suppose open neuroscience is going to be our focus, and ensuring that, again, we're seeking maximum benefits for the patients at the end of the day, who all of you are talking about, and minimizing risks to them. Jen, I was wondering if you could comment from a legal point of view on the topic of open science, but really I'm curious about everybody's input here.
I think I might leave it to Zelma and you to talk about the patent question, but I would mention that it's interesting that some of the laws around the world that actually already exist to govern NPD often in some of the cases include a data gathering or requirement. It's meant for a form of oversight, and there is a possibility to expand on that in a way that would help us to collect information in these cases to improve the research. So that's one possibility, is the amendment of those kinds of laws to actually build that kind of approach if that was felt to be advantageous.
So that's sort of a move that PCNEC could consider as it thinks forward, where it could take some action and have impact on policymaking, for example, including some of the regulatory systems.
So the registry, that's a great idea. It's a great way to do it. The problem is always the funding, right? So these databases all require some inherent funding, which is just not available through the channels that we have to us right now. So it's all very nice to be able to say we're going to do it, but it also has to be provided in such a way. And I know there are people working on this that And so there is potential hope for that down the road.
Helen?
Yeah, I was going to say, I mean, that's exactly it. I mean, I've watched Mike Oken with the Tourette's Consortium. He got some backing from the Tourette's Association to set up a registry because DBS for Tourette's is not approved. It's not, again, the US scene has got a whole other set of issues that you don't have in Canada, or you do. I mean, it's just regulated differently, but that, he was able to get funding, but it still is a hard cause, that small group is committed to doing it. The, Brain Initiative of NIH, had the forethought to say, gee, we'll fund a group to set up a master registry of all of the electrophysiology data, since everyone's collecting electrophysiology data. Well, they made a demand that everybody with a grant would be obligated to put data into somebody else's design database, and that nobody offered any money. Like, who's supposed to do that? And then worse, well, if it's in there, what's going to happen? Is this group going to be funded indefinitely? I mean, again, look, we all know, you have a job and you're required to keep your files for X number of years. It's like taxes. And the question is where does it go if you retire and decide you want to be a farmer? Or, what do you do with your stuff? I mean, I don't know. I don't know how to get out of what I do myself, so I don't have a solution. But I think this notion of like-minded people, you have a small community that can be thinking about what are elements that everyone could share, and you could actually get started. When it's so huge, it's just too overwhelming, and the activation energy is impossible. That's a kind of a downer comment, but sorry.
Well, I can end on a great comment. I want to thank PCNEC for, number one, starting this incredible forum. Number two, engaging with the Focused Ultrasound Foundation, because I think the intersection together is really interesting and necessary. And I want to thank all of the panelists and Dr. Mayberg for making the time to present today and share your thoughts and expertise. And so I'll let Dr. Ellis and Dr. Lipsman finish us out.
I'm also going to give my thanks to everyone for joining us today and let Dr. Lipsman have the last word.
Thank you so much, Judy and Susie, really, and Helen, for just an outstanding discussion and Q&A. I want to once more acknowledge the Focused Ultrasound Foundation for their terrific support, not only for this webinar, but for all the research across the board on emerging neurotechnology and the vision really going forward. I encourage everyone in the audience to really get involved locally and collaboratively on these issues. These are the key questions that really provide a lot of the context and a lot of the meaning for a lot of the work that we do, and help us define the priorities and next steps for our field. I encourage also everybody to check out our website, www.pcnec.ca, or to email any of us directly with your thoughts and follow up down the road. Thank you again, everybody, to all the speakers, everyone who joined us today. And on behalf of all the organizers, have a terrific evening, afternoon, and the rest of your day. Thank you.
Bye-bye.
Take care.